Hey everyone, we're a group of third year students. We're all currently studying biochemistry at the University of Otago.
We are here to share with you some of the groundbreaking research that's being done in the Biochemistry Department at Otago University.
Here are topics that we are passionate about, with each tackling their own issues in areas such as human health and environmental sciences.
We hope what you hear about today will encourage you to learn more about these issues and make your impact in the scientific community in the future.
I guess a little bit about me. My name's Georgina Wallace. I'm a full-time student, finishing my final year of my bachelor's degree in biochemistry, and I'm also part-time in the New Zealand Army, so I'm pretty busy. But if you told me I'd be where I am now, back in school, I would have laughed, because when I was in high school, I had no idea what I wanted to study. I guess all throughout NCEA, I did all the sciences, calculus, French and English, so I really kept my options open, which was really good because after doing my first year of health science, I really realised what I wanted to do, and I had the subjects to back me up when I found biochemistry, when it so aligned with what I wanted to study and what I wanted to progress in my tertiary education. I guess biochemistry is a pretty good hybrid between biology and chemistry, but in a ways that you can help with human health and disease by learning the intricate details of science, of how people work, and it really comes back to people, and that's why I fell in love with it.
Hi there, my name is Corey Gimlincy, and I'm also currently a biochemistry major at Otago University. Originally, I come from up north. I went to Hamilton Boys High School, and from very early on, I knew I wanted to come down to Dunedin, get away from home, and study some sort of science degree. I wasn't too sure what that was, so I chose health science first year to put off deciding for just one more year. Through that first year, I got exposure to the variety of science courses offered at the university and was able to make my decision to become a biochemistry major. So I chose to major in biochemistry primarily because of how the work done by biochemists can be used to help people and really make a difference. As a biochemist, you can work in many different topics, which allows you to work on something that you're passionate about and something you have a vested interest in. So that is how I came to end up studying biochemistry at the Otago University.
So one of the main projects currently being undertaken by the biochemistry department is breast cancer research. Cancer is obviously really prevalent in our society. It has been since the dinosaurs, and we know it will continue to affect our society into the distant future. Just to show you some statistics, it's currently the number one cause of mortality rates in New Zealand. Breast cancer in particular is one of the most common types of cancer in women.
Developing a cure for cancer is a very difficult task, as those biochemical characteristics that cause cancer are also what makes us human. Breast cancer research in the biochemistry department, headed by Dr. Anita Dumbier, looks to focus primarily on making a difference in the short term rather than long-term goal of curing cancer. The goal of this department is to find ways to treat breast cancer more effectively and at an individual level. The individualization of medicine is the way of the future if we want to be able to help everyone as effectively as possible. Everyone is different. We all have different genetics which have a strong effect on how any one of us might respond to a particular treatment, or how likely each of us are to develop any particular disease. In regards to breast cancer research, people who have been diagnosed with the same type of cancer may have quite different genetic predispositions to that form of cancer, and this can affect how they respond to a particular type of treatment. Anita's research looks to utilize biomarkers, markers within the genome, to identify which patients will respond well to standard cancer treatment, such as chemotherapy and radiotherapy, and which patients might respond poorly but will respond better to other targeted treatments.
Breast cancer is a really sampleable disease. This means we can take samples before and after treatment to see how effective they are. Clinical research, such as drug treatments, can be conducted between the time that the tumour was diagnosed and the time of surgery to remove that tumour. In New Zealand, the time between diagnosis and surgery is fairly large due to the unfortunately limited amount of physicians that are able to conduct tumour removals. This time before surgery, however, can be used to test different drugs and determine their effect on this tumour. Hopefully, some drugs will be able to shrink the tumour and make its removal during surgery easier. One project currently taking place in the biochemistry department is looking at a method using the immune system against the cancer. Analysis of the effect on this drug, called aletrozole, has shown that some patients respond to treatment and the tumours shrink, whilst other patients are non-responders.
Patients who respond poorly to the drug displayed an increase in the amount of immune cells within their tumour. These immune cells were thought to be having a negative effect on the body and were rather helping the tumour survive. The higher the expression of an inflammatory response metagene, the worse a patient responded to the anastrozole drug. A large set of genes that were being upregulated by the metagene were chemokines. Chemokines are a small protein that are secreted by the cells to recruit inflammatory cells to the appropriate site and elucidate an inflammatory response.
So this discovery actually prompted the question, could estrogen deprivation prompt the production of chemokines, and whether or not these chemokines were responsible for recruiting immune cells to the tumour? To test this hypothesis, an estrogen receptor positive breast cancer cell line called MCF7 was treated with and without estradiol, which is one of the primary estrogen hormones. Estrogen deprivation within these cells increased chemokine expression, and to test whether or not these chemokines were responsible for recruiting the large amount of immune cells to the tumour, a transwell assay was set up. This is pretty much the same as the prior experiment, whether this time a transwell containing a variety of immune cells was placed over top of the well containing the MCF7 cells. So the bottom of the immune cell well is a membrane permeable enough to allow the immune cells to pass through towards the tumour cells if they are stimulated to do so. This test showed that in the absence of estradiol, more immune cells migrated through the membrane towards the tumour cells, as you can see here. These tests supported the hypothesis that estrogen deprivation caused increased chemokine expression, which subsequently recruited immune cells to the tumour.
Hi everyone, my name is Isaiah Gapiga. I'm a third-year biomed student at the University of Otago. This allows me to study topics that I am passionate about, like cancer and human health. Throughout high school, I knew that I wanted to study biomed, but I didn't take papers that reflected that. I took French, English, PE, but I did take biology, which is where I learned about biochemistry, which started the longest love-hate relationship of my life. Studying biochemistry at the beginning was very interesting for me because I didn't take physics or chemistry since year 11. By the time I caught up, I fell in love, and through biochemistry, I'm able to study what I'm passionate about while also doing my part to tackle issues that affect everyone.
Hello everyone, my name is Raya. I'm a third-year biochemistry student at Otago University. A little bit about myself. I'm from Middle East, specifically from Oman. I have been always passionate about the environment and science, and I participated in many climate change and environment movement back home, and I have been accepted in the government scholarship program to study abroad, and they accepted me here in Otago University, so I have been here for 4 years now. I studied Foundation and English at the Foundation Center in the University of Otago. So yeah.
Life thrives in all corners of the globe, each organism tailored to their habitat through millions of years of natural selection. Each have found their own way to thrive in the harshest of environments, from the heat-ridden dunes of the Sahara to the sub-zero waters of the Arctic. Recently, research has been conducted by the University of Otago into the mechanisms behind how bacteria can survive being frozen in an iceberg, and animals like the fish of the Arctic are able to survive colder than freezing temperatures. The key to this survival mechanism is called ice-binding proteins. Freezing is a difficult problem to solve for most living things, because water is a key component of most biological processes. When water turns into ice, it forms in sharp crystal structures, which can cause damage to tissue and cells while also expanding. At the same time, it also causes an osmotic imbalance due to the change in solute concentration. Species that thrive in the harsh cold environments have learned to deal with freezing in several different ways. Some expel most of their water from their body, which causes them to dry out and not be vulnerable to freezing, while others use ice-binding proteins to prevent water inside of them from freezing, while others also use ice-binding proteins to control the freezing process inside of their cells. All of these mechanisms allow for them to circumvent the harsh consequences of freezing.
The biochemistry of this field is poorly understood. However, many researchers such as Craig Marshall are interested in studying the biochemistry behind these micromolecules. Ice-binding protein can interact directly or indirectly to the ice.
directly, such as ice nucleating protein or anti-freezing protein, which work completely opposite each other by changing the freezing point. It's known that the pure ice grows at 0 degree. However, if you left a pure water with no soluble to cool at minus 8 degree, some of the molecules will freeze and the rest will stay liquid for so long. Therefore, in order to form ice, you might decrease the temperature and weight, or make a nucleation center by adding nucleating protein. This protein has been used in cloud seeding to prevent dry land, and some bacteria that I don't know how to pronounce their name release this protein to freeze the plant tissue and damage it so it can suck the nutrients from the damaged tissue. On the other hand, the anti-freezing protein works the opposite way. This protein binds to the nucleation center and prevents from growing any further. This protein is the most known protein and studied. The third type of ice-binding protein is called recrystallization inhibitor protein, which does not change the freezing point and does not interact with the ice directly. This protein minimizes the annealing of ice, which basically make smaller pieces of ice instead of large pieces of ice.
Through the understanding of the structure, properties, and functions of ice-binding proteins, scientists may be able to tackle issues in agriculture, medicine, and climate change. In the future, there may be genetically modified organisms, such as livestock and crops, that express ice-binding proteins, allowing them to survive cultivation in harsher environments. There may be medicinal uses for ice-binding proteins, such as cryogenics. This could allow for long-term storage of a multitude of items such as body parts, sperm, eggs, and possibly full humans.
Understanding this type of protein can allow us to create a new way to survive climate change. Some studies show that controlling the expression of ice-binding protein will allow us in the future to achieve a higher freezing resistance of plants to survive the climate change. Because the unpredictable freezing in spring and autumns due to the climate change create a really serious issue to the plants and create imbalanced ecosystem.